Hello, everyone. I am pleased to present Dr. Stephen Berkowitz, Principal Scientist in the Department of Analytical Development at Biogen IDEC in Cambridge, Massachusetts. Steve's talk today will be on the use of the analytical ultracentrifuge. Steve?
Well, thanks, Paul, for the introduction. And today we'll be talking about the use of the analytical centrifuge to characterize very large, complex biopharmaceutical. And specifically, we'll be talking about characterization work on adenovirus, which is a viral gene therapy delivery system. I'd like to point out that before I really get into this, that in developing biopharmaceuticals, there's a number of pieces of information that we need all the time. That is homogeneity information, purity assessment, stability, consistency of manufacturing, comparability information studies when we make changes to our product to make sure we're making the same product, and information about potency. In this case, we'll be talking about chemical potency. So we need analytical tools to do this. And unfortunately for viral vectors like adenovirus, we run into some unique problems trying to get this information, and that's due to the size and complexity of these large particles. So basically what I'm going to try to do is show that the analytical centrifuge can actually play an important, helpful role in meeting these challenges. And the information that I'm going to be talking specifically is actually available in various periodicals, which are indicated here. This is published work, and we also have some information on Alliance ProteomeLab at their website, which is indicated here. So if you need additional information about this talk, you have sources for them. Okay, so before again start in getting into the detail, again I want to reiterate some positive attributes about the analytical centrifuge in characterizing biopharmaceuticals, especially large complex biopharmaceuticals like adenovirus. And that is, in most cases, we don't need to do any method development. In most cases, we don't need to do any sample preparation. And you'll see in this case, all we need to do is transfer the sample directly from the vial to the cell, and that's all that's going to be required. In terms of sample analysis, it can be done, obviously, directly in the formulation buffer, and that's very important. We don't have to change, such as maybe doing SEC, we're going to change the mobile phase. You can't use the formulation buffer. Here, we're going to be actually doing the analysis directly in the formulation buffer. In addition, there's mineral surface contact. There's only 4 walls or 6 walls that come in contact with the sample. 2 of them are the windows that are sandwiched in the centerpiece and the centerpiece itself. We also have enormous dynamic range in which we can use the analytical centrifuge in terms of molecular weight. And in particular, we're going to be looking at adenovirus, which is about 150 million molecular weight. So we're going to be looking at some very large particles and exploring the capability of the instrument to really characterize material at this very upper size range. In addition, there are a number of different detectors available, a UV detector and a refractive index detector, the rarely interferometer, which is really nothing more than a differential refractometer, and actually we're going to make very good use of that latter detector in this work. So for the next slide, actually for those who have never seen the modern analytical centrifuge, I actually have a picture here of what it actually looks like. It's about the size of a prep centrifuge, and this arrow actually indicates a little mechanical pencil so you get an idea of, in terms of perspective, the size. You can see the rotor sitting on the back on top of the centrifuge, a black object. There are 2 cells to the right of the pencil. Those are the actual centrifuge cells. And then to the left, you see the components that make up that cell. There's the 2 windows and the centerpiece. Now, if we open the door on the top of that centrifuge, that black handle, we roll it back, we can actually look down into the rotor chamber, and you can see the rotor sitting on the drive shaft. And this first arrow that's pointing to the left, that's indicating the source of the UV visible detector's lamp. So light's going to come up through that hole, and you'll see shortly that it goes through an optic bench, and then actually will then be used to illuminate the cell. The next arrow, which points to the right, I guess the first arrow pointing to the left, excuse me. The arrow pointing to the right actually indicates the location of the photo multiplier. It's sitting right underneath that hole. Right now there's no cell in there, and at the bottom of that hole there's a photo multiplier, and there's a slit image which moves across that photo multiplier and actually scans the cell. The arrow that's pointing up, that indicates the location of the rarely optical CDC camera sitting on the bottom. And you'll see in the next picture where it gets illuminated by the laser when we assemble the rest of the optical bench. So now the optical bench is inserted into the bottom of that chamber, and the black arrow indicates the laser light source that will then illuminate the cell and be detected by the CDC camera. And the arrows, the yellow arrows, indicate the location of the optical bench for the UV detector. So this gives you a picture of what the actual instrument sort of looks like if you've never dealt with one. Okay, in this talk we're actually dealing with three different types of techniques. We'll be talking about a little bit about the analytical sedimentation equilibrium and the cesium density gradient. This will be using UV detection, and here we're taking advantage of buoyant density to do the actual separation. We'll also talk briefly about band sedimentation velocity in a cesium chloride solution, and again we'll be using UV detection. And here the separation is basically involves the hydrodynamics of the mass and shape, but we'll also be taking advantage of the buoyant density effect. So these three parameters are going to play an important role in this technique. But the bulk of this technique and the major form of sedimentation, which we feel is most important in our application of adenovirus characterization, is classical boundary sedimentation velocity. And in this case, we're going to be using predominantly the Rayleigh interferometer, the refractive index detector. And again, this technique separates, again, predominantly on hydrodynamics, mass, and shape, but the role of buoyant density plays somewhat of a role here also. Again, this is just to reiterate the centerpieces for these different techniques. The one on the left, the centerpiece is a classical double sector cell. The arrow on the top indicates one of the filling holes, which allows you, when the cell is assembled, to actually introduce the buffer and sample. So the buffer goes on the left-hand channel. You fill the buffer reference material, a reference solvent on the left, and then you fill on a separate chamber the sample in the right-hand side. Now, this other cell centerpiece just to the right is a unique centerpiece that's specific to band sedimentation. And it looks just like the double sector, but it has a little hole. And in this case, we actually use that hole. That's where we place the sample. And the buffers that are used are then filled in the channel. We'll talk a little bit more about this, but just to show you, the centerpiece is uniquely different for band sedimentation. Okay, and lastly, this last slide before we really get into talking about some of our results, this actually gives you a surface model picture of the capsid of an adenovirus. It's roughly 90 nanometers in diameter, and it consists of hundreds of subunits. The most prominent is the blue little spheres on this capsid that have molecular weights approaching about 110,000. So just that one little subunit is almost the size of a monoclonal antibody, and there's hundreds of these little proteins that make up this rather complex virus particle. In addition to the capsid, you notice that this actually has little spikes. There's 12 spikes that stick out about 30 nanometers from the surface of the shell, and these are involved in the connection to the receptors on various cells where the virus makes contact and then is internalized into the cell. And the next picture actually shows you a cross-section of this to further indicate the complexity of the structure. Internalized within that capsid is the DNA, and in gene therapy, the DNA that's involved in the replication of this virus is deleted, and inserted in its place is the gene of interest that you're developing your gene therapy program with, and that gene is generally referred to as a transgene. So you can see that the internal cross-section indicates a lot more complexity. And one of the unique problems that occurs in making viruses is that when they get assembled, they frequently make what is called empty capsids. And that is the DNA that is normally present is actually absent in these particles. And that's one of the reasons we initially got interested in applying centrifugation, was trying to get a handle on trying to measure those empty capsids. The next picture actually shows you an electron micrograph of a collection of adenovirus, and those viruses that are completely intact show up as sort of like little white balls, as indicated by the blue arrow. However, those particles that are devoid of DNA, they allow the dye to penetrate and look like little shells. So these little shell-like appearance particles are actually the empty capsids, and they're trying to figure out ways to analytically measure the amount of that material in these preparations. In addition, we realized we also needed techniques for measuring and assessing aggregation. And that's an area that people have been interested in also, in trying to figure out what is the best way to actually assess that. Light scattering tends to be a technique that's very commonly used, but we're going to show you that the centrifuge can actually acquire this information at the same time in terms of getting information about the empty capsids. And lastly, we're really interested in understanding the native homogeneity of this material in terms of all the structural variants that are present. And again, we'll show that the centrifuge offers a significant capability of being able to provide a lot of detailed information about the various structures that are present in these preparations. Okay, so getting back to empty capsid, classically, these materials are removed during normal laboratory purification of virus or adenovirus by using preparative sedimentation equilibrium and a density gradient. That's a classical tool that's used very commonly in the purification of viruses. And when using that technique, which is a high-resolution technique that separates on the basis of buoyant density, you can actually get rid of these empty capsids. Because the buoyant density of an intact virus, an adenovirus, is about 1.3 to 1.35, whereas the empty capsids have a much lower density, about 1.29 to 1.23. So if we can use sedimentation equilibrium density gradient on a prepper level, we wouldn't have to worry about this. But the issue is when you're going to try to commercialize a product, a gene therapy, this type of purification route is just not economically feasible. And what people need to do is actually resort to chromatography, and that's what's commonly used in the purification of viruses. And unfortunately, that doesn't totally remove these empty capsid particles. So they're going to be present, and we need a way to measure their amounts. Well, why not use analytical sedimentation equilibrium and a density gradient and do that online? Actually, people have, Gary Velkamp at Sharing Plow has actually done preparative sedimentation equilibrium and density gradient, where you actually do this separation, take the whole tube out of the centrifuge and then manually fractionate it, have it go to a UV detector, and actually try to attempt to measure the amounts of empty capsids. And it can be done, but it's very awkward. Here, we can actually take the analytical centrifuge and do this directly in the analytical centrifuge. So you're actually doing the separation and then using the instrument as an analytical tool to make the measurement. So this next slide shows some work. This is done in collaboration with John Philo at Orion's ProteomeLab. John and I worked together for some time in this area, but John did this experiment in his lab. This is an actual analytical run of adenovirus in the analytical centrifuge, and we're doing analytical cesium chloride density gradient equilibrium. So what you do is we have two preparations. The data at the top is material that was adenovirus that was purified by ion exchange, and the material on the bottom is material that adenovirus material that was purified by preparative cesium chloride density gradient. And as I mentioned, using that purification approach, you don't get empty capsids. And clearly you can see that the material referred to on the top as EC, that's empty capsids. And we actually can see there's actually two species of empty capsids, and that's well known that there could very easily be a number of different species of empty capsids in your adenovirus preparation. So you do this, you start off with a solution that's at a uniform cesium chloride density, very close to that mid-range between these two particles. And you can see on the bottom, you start off with a very low level of your material. And then when you turn on the centrifuge, you're running this at about 15,000 RPM. That centrifugal field will actually redistribute the salt to a certain extent to actually establish a density gradient that's sort of implied by that dotted line on the top figure. So you get the actual density gradient that's stabilized, and the material in that cell will now migrate to the buoyant density, and you'll actually get an equilibrium band as we achieved here. And you can see in the case of the material that was purified ion exchange, we actually have the empty capsids, and we can integrate that area and actually quantitate it. Whereas the material purified by cesium chloride preparative density gradient, there's no material, no empty capsids. What's also interesting is that we've actually shown that there's actually different species that actually have buoyant density even greater than the actual intact monomer, and that's indicated by the material, by the HV, that's higher density virus particles. So there's several species here that we can already see getting additional analytical information that's sort of relevant to characterizing this material. There is other types of species that are more dense, more buoyant, or less buoyant than the intact monomer, and can actually get information about their presence through this technique. Well, as I mentioned, there's really some really positive key attributes about analytical sedimentation and equilibrium sedimentation equilibrium receiving chloride density gradient. It has this very high resolution in terms of discriminating particles on the basis of buoyant density. And that could actually give us information about the microheterogeneity of the structure, and also indicate potential damage that may occur to the virus. So this is a nice way of getting that information. However, there are some rather negative aspects to this technique that made us sort of think about some other things to approach to measure empty capsids. And first, it does take about a day or a little more to get the experiment done to reach equilibrium. You need to be very careful about getting the solution density correct. because you can easily be off, and the material may wind up like the meniscus, and you can't integrate it, or down the base. So you have a very narrow window of density gradient to work with, so you have to be very careful about making the starting density correct. You're also left with a sedimentation buffer that's very different than what you had in your formulation. So you need to be concerned if you wanted to get into some other information about the native structure of the various particles present. You're going to be concerned about whether that salt has changed anything. And we'll see examples of that a little later on. And one of the most sort of glaring problems, though, is that when you do this type of work with adenovirus, you generate a very sharp intact virus peak. And this intact virus peak is going to be pretty large in size in order to be able to integrate the small levels of empty capsid. So the sharp peak actually gives rise to nonlinearity and in terms of response of the detector, and that creates a little bit of a problem in terms of doing the quantitation. That leads to also sensitivity issues. So we started to think about, well, what can we do to broaden out that peak, and maybe we can apply band sedimentation velocity in the cesium chloride gradient. Now, this is another technique which I alluded to earlier. And the next slide actually, again, goes back and shows you the centerpiece. So this is a special centerpiece, as we talked about at the beginning. On the left, you put your buffer. The right, you put normally your sample, but actually you put the reference buffer again. So both these chambers, when you do band sedimentation, are filled with the same buffer. One's going to be acting as your blank or reference side, your left, whereas the right is actually going to be the material, the virus particles that are sediment through. And you basically put your sample about, say, 15 microliters in this hole, and you then assemble the cell, fill the buffer, and the buffer level has to be below this channel, the bottom channel. Otherwise, when the virus gets layered out, it won't actually effectively get layered onto the liquid, the cesium chloride. So the amount of liquid you put into these channels has to be such that the column height is below that channel. And what you do, you turn this on to a low speed. The sample that you place in that well actually gets layered out onto the cesium chloride, and you actually get a band formed. Now, there's some critical aspects of doing band sedimentation, and one of the critical aspects is the solution of your virus has to be in a solution of lower density than the solution you're going to be sedimenting through. And in this case, we're going to actually be sedimenting through a rather high concentration of cesium chloride, so that doesn't become much of a problem. So this is sort of the basic mechanics of what's going on here in terms of doing the band sedimentation. The next slide actually shows you an example, and this is material. You're looking at band sedimentation cesium chloride solution, and the actual density is about 1.29, so it's very high density, but sufficiently low enough to allow this material to actually sediment to the bottom. But during that, we'll get separation of our intact material from an empty capsid. So what you're looking at, this is a virus preparation which was made in the lab and was purified by cesium chloride, and it doesn't have any empty capsids. But we're looking at the progressive scans during the experiment that are overlaid in this picture. And you can see the band just migrates toward the bottom of the cell, and as it does so, it broadens out, and as it broadens out, it lowers its peak maximum. So it looks like a very nice a picture of the progress of the actual sedimentation. Now, if we go to some examples where we have purified material by ion exchange, and we're looking at a single scan, we're not following it in detail. We just capture one of the scans sort of mid-range into the run. So the first two top figures are, again, adenoviruses made by ion exchange, so therefore they contain some empty capsids, and that is again indicated by the ECL peak on the top. And in the preparation in the middle, we actually have two empty capsids that we can see, two different species. And we have intact virus monomer peak that's close to the bottom. Now, and then the bottom trace actually is an adenovirus preparation that was purified by cesium chloride. So that doesn't have any empty capsid. You get this nice single peak. In both cases, other than the empty capsid, that's all we're seeing. What would appear to be is the monomeric intact virus peak and the empty capsids. And you can see the empty capsids are very nicely resolved away from the intact monomer peak. We can do some work where we actually put different amounts of material into the well and do different experiments. So we want to see what is the quantitation in terms of linearity. So with different amounts of virus in the well, and then looking at the area, you can see we get nice linearity. So this actually overcomes band sedimentation and cesium chloride, actually overcomes some of the issues we mentioned in terms of sedimentation equilibrium and cesium chloride density gradient, and that's particularly in terms of the nonlinearity of the technique. And we've also significantly reduced the amount of material you need to do this band sedimentation experiment compared to the amount of material and sort of more difficult setup that you have with the equilibrium approach. However, again, we're still doing the measurements in a buffer system that's very far removed from the formulation buffer. Oh, and in addition, one of the other sort of aspects of band sedimentation that you need to deal with in terms of getting good quantitation on empty capsid is you need to accurately sort of assess the extinction coefficients of these different species, because they actually do have significantly different extinction coefficients. If you don't take that into account with UV detection, you're going to get an answer that's not going to be correct. Well, if we could use refractometric detection, such as the Rayleigh interferometer, we can actually overcome this problem. And that's indicated by this little table. And what we have is there's two rows, the protein and DNA, and the second column indicates what's called the refractive index increment. And that is a term that's very much analogous to extinction coefficient in UV detection, but it applies to refractometric detection. So it's a parameter that relates to the response factor in terms of refractometric measurements for, in this case, protein and DNA. And you can see the DNVC for those two different chemicals, which are the main constituents of the adenovirus, they're not that far apart. However, if you go over to UV and look at what they might be at 260 or 280, the wavelength you use to do a UV measurement, you can see that both these substances have very different extinction coefficients. And that presents a problem, as I mentioned to you, if you don't take that into account, because the empty capsids are clearly just protein in nature typically, and the intact virus has a significant amount of DNA, so they have different amounts of the extinction response. And if you wanted to actually calculate the DNVC and the UV extinction coefficient for the adenovirus, well, we can do that fairly simply by using this little equation, which just upon additivity, knowing the amount chemically, how much DNA there is and how much protein, we can basically use these pieces of information in terms of extinction and calculate the extinction coefficient for the virus in terms of refractometric by computing the DNVC, or actually computing the UV extinction coefficient. And if you look at the refractometric measurement, you can see that there's hardly any difference in the response factor between the adenovirus and the protein. They're almost the same. Whereas again, you look at the UV detection extinction coefficients, you see that there are significant, very large differences, and still you're going to need to deal with making some correction. Well, unfortunately, you just can't apply refractometric detection to band sedimentation because you have such a significant salt gradient in the run that it actually overwhelms the signal from the actual material of interest, the adenovirus. So for both these two techniques, sedimentation equilibrium in a cesium chloride density gradient, or band sedimentation in a cesium salt solution, you can't use refractometric detection. Nevertheless, we can actually use this detector system to do classical boundary sedimentation. And that's what we'll be talking about next. So if we can actually do boundary sedimentation, velocity sedimentation, and use refractometric detection and get this successfully done to be able to measure the empty capsid and the other particles that you'll see present in these preparations, we'll actually achieve some very interesting things. We'll be able to do the measurement without any sample preparation. We can just fill the centrifuge cell straight from the vial into the cell. In addition, we don't need to take, as we mentioned before, into account the difference in extinction coefficient because their differences in terms of refractometric measurements are very small. The sample, and most importantly, is now analyzed directly in its native state in the formulation buffer, and hence we can actually measure meaningful aggregation and assess the sample's homogeneity in terms of microheterogeneity that may arise from the assembly intermediate or damage to the structure during the actual production. In addition, we'll be able to actually measure very accurately simply total concentration measurement from the same run. We'll also be taking advantage of the modern Rayleigh interferometer, its automation, high sensitivity, which through specially designed software can actually almost approach the detection limit of UV detection. The other point is this type of detector has very high linearity range. You don't run into non-linearity at low concentrations, although that's not a very major factor, something I'd just like to point out. The next slide actually shows you sort of example of the boundary sedimentation experiment. And this is not a refractometric trace, it's still a UV trace, but I'll just give you an idea of what you're actually looking at. You're actually getting these scans at different times, and we have them overlaid, and you can see the position of the reference meniscus which is the furthest pointing down, the furthest signal on your left-hand side. Then you see the sharp spike from the sample meniscus. And then you can see each of these data scans that are obtained. They represent a sigmoidal curve in a flat area. The flat area generally is referred to as a plateau, and then it goes all the way down to the base indicated on your right-hand side. So you're going to see an overlay of these traces. In this case, you're looking at UV, but we'll be looking at refractometric traces shortly. So each scan is taken at a particular time, and we're looking at the signal at different radiuses, and that gives us this sort of signal corresponding to S, which is a function of radius, which is the signal from the detector at different radial positions at a given time. So this is what classical boundary sedimentation data looks like. And I should point out an additional benefit when we use refractometric detection that's rather unique to using low speeds. And so if you're working with adenovirus, we don't need to go very high because this material is very large, 150 million molecular weight, 90 nanometers or so, if not a little bit bigger in diameter, and we need very low speeds. And one of the things that you need to pay attention to classically when you use refractometric detection in the centrifuge is you need to make sure your sample buffer matrix and your reference buffer matrix are actually matching. If you have difference in salt concentration, you're going to get differences of signal coming from just the sedimentation of the salt, because at high speeds, typical with like monoclonal antibodies, you're actually going to get significant sedimentation of the salt, and that's going to give rise to a refractive index signal that's going to overwhelm the signal of your material of interest. Similarly, you should also try to strive to have the volume of the reference buffer and the volume of the sample buffer to be the same, so the meniscus are pretty much matching. If you have a mismatch in the meniscus, you can get a differential sedimentation of salt just from the mismatch of the menisci. And these things need to be paid attention to normally in refractometric detection in the ultracentrifuge, but for adenovirus, we're going to be at 3,000 RPM. That's one of the lowest speeds you can go with this instrument, and it doesn't become a factor. At such a low speed, we're not going to get any significant sedimentation of the salt. So these things that you normally have to be concerned with are not an issue, and that further simplifies the application of using refractometric detection in the centrifuge. Okay, so let's take the next few slides and just take a moment to sort of talk about the analysis end of this. And this is actually a pretty complicated subject, and I'm just going to touch upon it very briefly to just give you a feel for what's going on. In today's modern approach to analytical ultracentrifugation in terms of classical boundary velocity sedimentation, people are now making use of what is called the Lamb equation. And this is an equation that was developed back in 1929 with Dr. Lamb, who was a co-worker of Svedberg in the early days of development of the analytical centrifuge. And this equation basically is a theoretical equation that allows one to calculate the sedimentation concentration profiles, those data scans that I actually showed you. You can actually calculate those scans for a single component with a sedimentation coefficient S and diffusion coefficient D, and as a function of radial distance and at different times in the sector cell for an ideal solution at a specific angular velocity or rotor speed. So for a single component that's behaving ideally, knowing its diffusion coefficient and sedimentation coefficient, I can actually calculate these scans that I showed you. And that's now applied today to solve or to analyze the data from classical boundary sedimentation. Although this equation has been known for a long time, there was no specific solution to that equation. But today, we can now take advantage of numerical analysis techniques that can be solved with a simple laptop computer or desktop computer. And these things are now available, and we can actually model the entire sedimentation process with these using the appropriate computer program. And the particular program that we're going to be talking about in this work is a program called CsCl, developed by Peter Shook. And at the bottom, I've indicated the website for Peter Shook. And if you're interested, I think you should go to that site. And it's just chock full of information about how this software works and all the details. And it's really a great source of information to get an understanding of what's going on in this program. I'll give you just a little smattering with these few next slides. So what we're doing is basically solving the Lamb equation. And in this case, we have a simulation of adenovirus solutions. In the left-hand side, we have monomeric virus particles, and the top simulations are for a UV detector which typically has a signal-to-noise ratio of about 100, whereas on the bottom we have refractometric traces of the same sample. But given the higher signal-to-noise capable on the refractometric, about 1,000, you can see the better quality data you can get with appropriate concentration. Needless to say, you have the left sort of panels for both detectors. This is what you would get by using Centrifugation to simulate for a homogeneous virus solution. You actually can compute the data and get these traces, and you could actually compare this back to your experimental data and see if it made sense that the material is only monomeric. However, if you go to the next set of panels in the middle, we've added a simulation where we have a 5% contamination of dimer. And since this is a spherical particle, we can actually very easily calculate the sedimentation properties of the dimer, and we can do the simulation to see what the data would look like if you had a sample with 5% dimer. And the ones on the to the right, those two panels, it's a more complex mixture containing 3% dimer, 2% trimer, and that should be 1% tetramer. That's a mistake there. It should be tetramer. So this is a more complex sort of solution. And there's a couple things I want to point out is that if you go back to the monomeric situation, the plateau you can see is very flat, and that's very diagnostic that there's very low amount or no significant amounts of aggregate. However, you go to the more complex situation where you have various components present, and you look at that plateau region, you can see that that plateau is starting to tilt down. And you can see, in the case of the refractometric, you can actually make out the unique boundaries of these various components. But if you go up to the UV, where the signal-to-noise is not good enough, they all sort of blend into each other. But needless to say, you can see the tilting of that plateau region. Again, that's very diagnostic, a visual diagnostic that your sample contains heterogeneous material or aggregated material in this case. So what we're doing is actually use this program. It can calculate model data, and what we want to do in using it is to then basically take the experimental data and use the calculated data to do the modeling. Okay. So what we have is, on one side, this S term, that's your experimental data. That's all your data scans. And each data scan really is a summation, as they have here indicated as a summation as an interval of all the components. The S term is the Lamb equation. So each of these data scans is a collection of different species, and each species has a Lamb equation solution. And what you want to do is find out is how much concentration of each of that species is present. So if you multiply the concentration of that species times the solution to the Lamb equation for that specific species and sum them all up, you should get back the data. So that's what the program is doing. It's going to if you go on to the extreme right-hand side, it's trying to numerically sort of solve or match the data by basically summing up these terms. So in using the program, you actually contain the number of components that you're willing to sort of investigate. Typically, anywhere from 50 to 250 components you say you're willing to look at, and then you define the sedimentation range over which you're going to look at species. And you then do this calculation internally in the program. It actually will search out the solutions to the Lamb equation, and then search out the best matching concentration for each of those species, sum up that term, and then look at the experimental data. In the next slide, you can see this is becoming the criteria. So you have your experimental data, the S terms, you have all that data that you've collected, and then you have your calculated data that you do. For every scan, you basically come up with a model, and you have to reproduce the data for every one of those scans that you've taken during your experiment. So you want to match the difference, minimize the difference in the experimental data and your calculated data. So what you want to do is minimize the square of the deviations, and you want to basically drive that to zero. Well, in doing that, you want to also figure out the concentration of each of those species that you're saying are present in the solution, and that's done by nonlinear least squares analysis. And it may sound a little bit complicated, and it is complicated, but if you get a general gist of what's going on, I think you'll find that this is an extremely useful program, and people today are really making good use of it. The other point I'm making in minimizing this difference between the experimental and the calculated data, or the theoretical data that you're calculating, you can't minimize perfectly to zero. You can't drive it to a perfect zero, because what happens is there's noise in the data, and if you try to minimize the noise, you're actually going to create artifacts. So you have to minimize, but you need to not overfit the data. And there's a tool that's built into the program that involves regularization. That's another mathematical tool that actually tries to minimize the amount of fitting, but enough that it really matches the data as best as possible without overfitting the data, in which you start fitting the noise. So there's some built-in capability to avoid this situation. But in essence, this is what's sort of going on in the program. This is a very high-altitude view, but just give you a somewhat taste of the difficulties that, or the complexity, I should say, that's going on here. But it's very doable, and you get very successful results from this. So the remaining talk, we'll actually look at some data. And this is an actual refractometric set of scans that was taken from a preparation of adenovirus that was made by ion exchange. And right away, if you look, we can see some unique features. You can see that we're developing a slow-moving boundary as we get near the base, and that actually corresponds to the empty capsid. Another interesting aspect, as I pointed out earlier, is that if you look at the plateau, you can see it's fairly flat. And again, this was very diagnostic that the amount of aggregated or larger species is very low in this sample. So we take all this data and process it through the SedFit program, and we get what is basically a distribution of sedimentation coefficient plot, which looks very much analogous to a size exclusion chromatography run, only it's running in the opposite direction in terms of how the big things come out later and the small things come out early. So you can see that this data has been transformed by this program to give this distribution of sedimentation plot, and you can integrate it so that the main peak is the large peak, that's your monomeric material. The material to the left corresponds to empty capsid material, and all the material moving faster than the monomer is basically what we need apparently aggregated material. And I should point out, and you'll see from a couple other slides coming up, that there's material migrating in the area of about 500 to 600 x, which is just to the right of the main peak. And you need to pay attention to that, because that's actually going to be very interesting what we think that material is, because it doesn't appear at this point, you'll see further, that material may not actually be aggregates, but it might be another form or a mixture of different monomeric forms, and we'll talk about that later. So anyway, you integrate these distribution sedimentation coefficients, and you get information on the amount of empty capsids, the aggregated material, and intact monomer. Now, if we take a look at another preparation, this is a different preparation, again purified by ion exchange, and again we can see some interesting features. I guess the arrows are missing here for some reason, but anyway, you can see that slow migrating boundary near the bottom of the traces on the left-hand side of the raw data scan. But also, if you look again at that plateau region, now you notice right away that it's actually tilting down, and that's indication that there's significant amounts of aggregated material. And this data is then put into the program, transformed to get the distribution sedimentation coefficient. And now we can see, yes, there's the intact monomer peak. And look at the empty capsid region, we actually now see two species again that we had seen in the cesium chloride density gradient equilibrium runs and band sedimentation runs. And we can also see that there's more aggregated material. We just integrate these areas, and we actually come up with the amount of empty capsid, aggregation, and monomeric material. And again, in this case, you can see there's a fair amount of material very close on the right-hand side between the 500 and 600 S area that is present. And just about all these preparations, we see this material. And again, we'll see later that this material may not be aggregated material, but again, might be something related to an altered monomeric material. Okay, there's two more preparations. Next one. This is actually another preparation, again, made by ion exchange, and it actually shows a somewhat different pattern. Again, the arrows are not showing up, so I'll have to explain it again. So if you look in the area where the empty capsid, you can see now that it's very broad, it's very tailing, and that there's also another boundary that appears at the very beginning of where the traces are. And that's a very slower migrating boundary that shows up. But again, you're looking at the plateau region, very flat. So this material should have very little aggregation, but now when you process it through SedFit, you can see the area where the empty capsid runs is very heterogeneous, and that's indicative of this very broad tailing boundary that we've seen in the sample on the raw data scans. And then there's a peak of fairly low sedimentation value, relatively speaking, compared to the intact virus particle around, I believe, something on the order of 20 or 30 S. So this preparation must have some broken up particles of different sizes, very heterogeneous. and that may come about through the empty capsids breaking up. The empty capsids, I should mention, are fairly fragile, and they are prone to breaking up into pieces. Of course, the intact virus itself may have been damaged. Needless to say, we can see this very different profile in this preparation. Okay, the next slide, which is I think the last one of this type, this is a preparation of adenoviruses made in the lab using cesium chloride density gradient. So this material is free of empty capsid. But if you look at panel A, this is the refractometric traces running the boundary sedimentation run of this material. And this material is actually stored at relatively high concentrations, around 10 to the 13th particles or maybe even greater. And when we took this out and diluted it down into the range to run the experiment, you can see that it actually gives us a very strange pattern. And where the blue arrows, you can see that at the beginning there's this very broad polydispersed indication of material that's very big. And if you take the beginning scans and process the data, you get the panel at the bottom, B. This is the sedimentation distribution from those beginning scans that we take. And you can see that the bulk of the material is now sedimenting somewhere between 10,000 and 1,000 S, whereas the normal monomers sediment at about close to 500 S or 450 S. And we actually even have material going up to 40,000 S. So it's very polydispersed and very large. And now if we take the remaining scans, and you can see, going back to panel A, we can see these actually unique boundaries that appear. And we take these later scans and process and we get panel C. And here we get these unique peaks showing up. The first one, it corresponds to the monomer. And we believe the second one corresponds to the dimer and so forth, trimer and tetramer. But if we pay, we're going to come back in the next slide to look at this monomer dimer and their positions, their sedimentation coefficients, and you'll see this plays out in trying to understand the material that actually sediments between 500 and 600 S. But anyway, let me just continue on with this slide. If you take this material and then put it into a band sedimentation in cesium chloride, which we had shown examples of before, so now we're sedimenting through high concentration of cesium, we get virtually one peak with a small indication of an aggregate, as indicated by, I believe it's VA there, virus aggregate. So this very heterogeneous and polydispersed material now looks fairly homogeneous. So again, this is indicative of the fact that changing the buffer matrix has a tremendous impact, and apparently the cesium chloride actually dissociates these aggregated materials, which are just tentatively aggregated, and when you run it in this band sedimentation, it looks like all the material is fairly monodispersed with a small amount of aggregate material. But clearly, the material in the vial is extensively aggregated, and that we can pick up by classical boundary sedimentation. So this illustrates a rather important point of trying to maintain the analysis in the formulation buffer. Now let's go back and look at this monomer dimer sort of situation. In the next slide, we have a table. And basically, it's a table which we can actually compute the sedimentation coefficient. This is a sphere. Adenovirus can be very well modeled as a sphere. So in the case of a dimer, there's only one unique configuration for a dimer, as indicated by those little balls. There's only one way you can put a dimer, and we can calculate the actual theoretical sedimentation coefficient of that dimer, and we can ratio that to the monomer. So knowing the monomer is, in this case, 443S, and from theoretical calculations for a spherical particle with this kind of sedimentation coefficient, the ratio of those two components, the dimer sedimentation over the monomer, should be a factor of 1.5. And if that's using the 1.5, apply that to the monomer, you multiply 1.5 times the monomer sedimentation coefficient, you should have a sedimentation coefficient for the dimer of 665. And if you look at the experimental data on the bottom, you actually see an excellent match. And if we sort of apply the same kind of reasoning to the higher species, trimers and tetramers, we can actually see, and it gets more complicated here because there's different configurations. The actual trimer at least can be put into two different configurations, if not more. Similarly, for the tetramer, there's at least three and probably more. And you can calculate these sedimentation coefficients for these various configurations. And if we just took the average, you come up with what is a theoretical sedimentation coefficient for the trimer and the tetramer, and you can see actually it matches fairly well to what we experimentally observed. But there's no ambiguity with looking at the monomer dimer. There is a very nice match. So the question now becomes, what is the nature of the material that's sedimenting between 500 and 600 S? Clearly, the smallest aggregate is sedimenting beyond 600 S. So we're seeing these preparations having a lot of material in this region. And our best speculation at this point goes back to the beginning of this presentation where I showed you a picture of the adenovirus with those spikes. And those spikes, which if were lost, would have a significant impact in making the particles actually move pretty fast. Although the change in mass is very small, the hydrodynamics would be reduced significantly so that they would have a smaller frictional coefficient able to migrate faster. So we're speculating that material in the 500 to 600 area, sedimentation coefficient area, is probably intact monomeric species that might be missing, most likely, the spikes that are on these particles. And that's also consistent with the data from the sedimentation equilibrium runs done in cesium chloride, because missing protein, you now have particles that have a higher buoyant density. So that might, again, go along with when we look at and characterize these preparations, and we actually do see material with buoyant densities higher than that of the intact virus. Now, this is speculative, but this begins to indicate sort of the capability of what the tech can do, can start giving you a very detailed picture of what you have. So, anyway, let's pick up the last topic of our presentation, which basically deals with measuring concentration. And as I mentioned, the way typically concentration is measured by simple UV measurements. And that's the way we do that with almost all our biological products, measuring UV. But with adenovirus, you run into a complication. So not only when you measure UV do you measure the true chromophores from proteins and DNA, you're also measuring apparent absorption from light scattering. So the monomer itself will significantly scatter light, but more importantly, whatever aggregates are present, they scatter light, and they will scatter light depending on their size and shape. And that leads to a lot of variation in what you might get for the same amount of mass material that's in different, has different amounts of aggregation. It's going to give you different apparent UV measurements. So back in 1968, Mazel and co-workers came up with a clever approach of basically measuring adenovirus concentration by denaturing it. By putting it into a solution of SDS and heating it, they knock down the material so you disperse it into its basic units, and that was the source of material you can make your UV measurements on. That followed up with Mazel, and then using this approach came up with an extinction coefficient, which you carry out your sample processing in the presence of heating it in the SDS, and then you do your UV measurement, and that extinction coefficient is based on the composition of DNA, the molecular weight of the DNA, and allow a protein measurement to get the amount of protein for a given amount of ODs measured via this process. So that's classically a lot of measurements are done to assess concentration. However, there's been a lot of difficulty in reproducing the methodology, and people have been concerned about what, are you getting the correct OD readings from this processing? And a group was set up about several years ago. It was called the Adenovirus Reference Working Group. A number of companies in collaboration with the FDA tried to come up with a lot of adenovirus which would be used as a reference standard. They would determine the amount of ODs in that sample and therefore assess the amount of virus particles per ml based on Mazel's extinction coefficient. So they developed this reference standard, and that reference standard is commercially available. Okay, so what we're going to do is, since we're using refractometric detection, we can now basically, as I showed you earlier, that we can actually just by measuring the change in refractive index, which is related to what is called the change in fringes, because the Rayleigh interferometer actually, which does give you a detection of the refractive index, but your measurement is in measuring number of fringes, times a constant, and you see this constant is the wavelength of the laser divided by the length of the path length of the cell. When you basically measure the number of fringes times this constant, you actually get the total change in refractive index of the sample. And when you divide that parameter by the NDC, which we showed we measured or determined by data earlier in this presentation, we can actually get the concentration. And that can be accurately done. And the table below, these are like six different runs which were done with this adenovirus sample, and you can see the reproducibility of the total concentration is fairly good. Within about 2%, we can reproducibly measure the concentration of the adenovirus. And at the same time, we can get information about aggregation and empty capsids. Although you can see that in the case particularly with the empty capsids, there's a lot of variation, and there's some difficulty about the stability of these empty capsids. And this material is actually in about 2.5% glycerol. That may not be perfectly good enough to stabilize these empty capsids, and may be one of the reasons for some of the variability, because these empty capsids are pretty unstable. If you look at the next slide, this is a material that we use in our company, which we measured many times, similarly as we measured the arm sample, the reference material. Again, you can see the data at the bottom, very good reproducibility of measuring concentration. And if you look at the amount of, look at the empty capsid, you can see the data here is much better, although there's higher amounts of empty material in this preparation. We're using much higher concentrations of glycerol, and that might be a factor of stabilizing these structures and able to do a better job of reproducibly measuring them. Although the aggregation you can see is very variable, that again may be a factor of the instability of these aggregates. So that's a problem that a physical solution property parameter of these aggregates. And as I showed you earlier, some of these aggregates can very easily be dissociated, and that may play out in some of the variability that we're seeing in the aggregation. So getting back to the arm sample, we measured the total concentration, and this little equation here, we can now convert the concentration of virus particles to actual virus particles per ml. So we can change the milligrams per ml material to virus particles per ml by using this little simple equation by taking the concentration C0 of the sample and dividing it by the virus molecular weight and multiplying it times Avogadro's number. Will actually then transform this concentration into virus particles per ml. And so doing, we're using more modern data, which is 15% DNA composition, and we're using we indicate a molecular weight of the DNA to calculate the actual total molecular weight of the virus. So with this information, we can get to the virus molecular weight, which is about 150 million molecular weight. And this is the number we come up with, 6.2 times 10 to the 11th virus particles per ml for this adenovirus. Now, the Adenovirus Working Group came up with a number of 5.8. And again, this is based on Mazel's extinction coefficient. But when Mazel developed that extinction coefficient, he based it on 13% DNA, as opposed to what is believed now to be a more accurate number of 15% DNA. So if we actually correct this earlier value, which was obtained by the ARMS Working Group, and correct it, and then average it with this value we obtained by the Centerpiece using refractometric detection, we get a value of 6.4 times 10 to the 11. Actually, the values are pretty close, but we think that one of the major differences here is in the DNA composition. In the earlier measurement by Mazel, implying 13% versus 15%, which is now believed to be the more accurate number. So, in fact, we get some actually fairly good agreement with the arms. And we believe now with two independent assessments of the concentration, we have a more accurate value of the arm concentration. Now, this kind of information can be obtained at the same time we're getting information in terms of empty capsid and aggregation. And that's indicated here, the advantage of using centrifugation to do the characterization work on one experiment under native conditions, under formulation conditions, with no sample preparation, no real method development involved. We can get information simultaneously on empty capsids, the amount, their solution properties, as we've talked about, the aggregation information. We can get information about the amounts and their solution property behavior. And we see we have some very different behavior that we can actually uncover in these materials. In addition, we get information about other structural aspects or heterogeneity that may exist in these preparations in terms of the amount and their solution properties. And simultaneously, last, we can get the total concentration of the sample without any concern of processing the sample. We're just running the sample directly out of the tube, out of the vial. We don't have to do any sample processing. We don't have to worry about light scattering. Light scattering is not a factor in refractometric detection. And UV contaminants, it turns out actually it was interesting when the ARMS was developed, the preparation, they actually found a UV contaminant. Fortunately, it didn't make much contribution to the UV measurement that the ARMS groups was gathering, but they did find a UV contaminant, and that would have at a significant level, if it had occurred, altered the results. Here, you don't have to worry about that because UV contaminants would not be part of your measurement. They wouldn't sediment. So, with that said, I think we're our next to last slide, which then makes a little comparison to some of the other techniques that are used predominantly in terms of assessing aggregation. Light scattering is a very commonly used technique, particularly dynamic light scattering. However, when you do light scattering, there is this problem with sample preparation, is what is called the dust problem. Particulate matter does have to be removed, otherwise it could contribute significantly to the signal. Although for adenovirus, which scatters so much light, it may not be as big an issue in this case. The technique is very low resolution, In terms of dynamic light scattering, the amount of information about size distribution is very low resolution compared to what you get out of sedimentation. Also in light scattering, it's very difficult to see the presence of low molecular weight material when you have lots of very large material. So preparations where we saw the presence of low molecular weight species, you would be very hard-pressed to see that in light scattering because the signals from the large particles would dominate. And I should also point out, in doing dynamic light scattering, you're using actually the same mathematical techniques which I alluded to earlier in this presentation are actually applied and utilized in dynamic light scattering. So although we see a lot of difficulty and complexity with analytical centrifugation, at least modern analytical centrifugation today, you have to realize those same techniques are being applied in dynamic light scattering. So if one is willing to do dynamic light scattering, one should be more than willing to do the analysis by a modern ultracentrifugation analysis today with these newer software programs that are readily available on the internet. In terms of field flow fractionation, which is now being looked at to some extent as another technique, you have to, again, in this case, be worried about interactions of your material with the membrane, and that membrane is porous, so there's a lot of surface area, so interactions are a concern. Also, during the measurement, there's large changes in concentration, although one thing for sure, the analysis for field flow fractionation is much simpler. And lastly, I should mention that both light scattering and field flow fractionation can be automated, so they're amenable to high sample throughput. Certainly a very positive aspect compared to analytical centrifuge, where you basically one run per day is probably pretty common, and with an 8-hole rotor, you're left with about 7 samples per day. So, with that said, I can conclude, and just want this last slide to thank a number of co-workers who have helped in various ways during the course of this project. So let me end this talk and field any questions that we may have.
Okay, at this point, I'd like to open up the floor to any questions that you may have.
And at this time, I'd like to remind everyone, in order to ask a question over the telephone, you can do so by pressing star and then the number 1 on your telephone keypad. Once again, that's star 1 to ask a question. We'll pause for just a moment to compile the Q&A roster.
Let me ask Steve. There's a question that came across the chat window from Andrew Timms. To your knowledge, has anyone used AUC with purified prions?
I'm not sure. I don't have knowledge about, so that's my unfortunate simple answer to that. Although you need to realize that the centrifugation technique can be used for virtually any material. As I mentioned early on, there's a very large dynamic window of sizes that can be analyzed. Believe it or not, people have looked at materials as small as sucrose, and materials up to micron size have been actually analyzed in the centrifuge. So there's no limitations in terms of the size given that window. So whether somebody has applied this to prions, I don't know for sure.
Okay, but it sounds as though it's theoretically possible.
That's right.
Are there any other questions over the phone, perhaps?
I'm showing no telephone questions, sir.
Okay. Let me ask one myself, and this has to do with aggregation. Steve, what percent aggregation seems to be of concern to folks?
Well, that's a good question, Paul. I think a lot of people in the industry are really looking at low levels of aggregation. Typical specifications are below 5%. Generally, when you get down to 1 or 2%, there are some issues, and I think there are several publications that have appeared that people are concerned with the lower end. So when you get down to 1 or 2% and going lower, the accuracy is somewhat in question. Although Peter Schiff, who's developed this program, made initial claims that you could go down to 0.1 or 0.2%. I think there's a lot of interest in trying to understand really how low one can go. And generally, anything over 1 or 2%, I think you have a good shot of getting reasonable information. But going below that, I think there's a growing concern about if one can accurately measure down below that and what it takes to achieve that. So it's a little bit open going below that sort of level of 1 to 2%, you might say. But there are claims that you could go down much lower, and I think more work needs to be done to make people feel comfortable that one can approach those lower levels.
Thank you. There's a question in the chat window from Jeffrey Miller. How does the presence of impurities affect analysis with regard to size and amount of virus?
Okay, well, In terms of refractometric detection, it sees anything. And that's one of the nice things about refractometric detection. It's a universal detector. So if for some reason you do have some other impurities that are falling within the size range of material that you think corresponds to adenovirus, well, you can't tell if it's adenovirus or it's some other impurity. However, one of the nice things about the centrifuge is that you do have a UV detector in addition to the refractive index detector, and that ratio signal might be useful for you to discriminate whether that falls within the line of this might be an impurity or this might be some other contaminant. That's a problem that you need to grapple with. So if impurities are low molecular weight, they're not going to sediment, so you're never going to see them as a boundary, you might say. They can be present, but they'll never be detected by the detector. So most of the small molecular weight problems are not going to be an issue. It's when you get impurities that have nothing to do with adenovirus, that are in the size range that are of interest to you, then the detector can't really discern them. But you do have some capability, if you run both detectors, you might be able to apply some discrimination to say that might be an external impurity not related to adenovirus. But that is a potential problem.
Thank you. Another question from Malcolm Madison. Can you tell me how you decontaminate the cell components? Do you worry about damaging the sapphire or other components?
Okay. That's a big question, and it's, again, rather unique to adenovirus. When we assemble the cell, when we fill the cell, we actually do this in a containment area. So this is in a laminar hood. We fill the cell, and then wipe off the cell with appropriate disinfectant, and then seal the cell, and then take it outside of that hood and put it into the centrifuge. After the experiment, we take that cell back out and go to the laminar hood and disassemble it and wash the material in a detergent solution. So basically, the materials are initially cleaned in detergent solution to begin with, and from the beginning and to the end. So there's a cleaning process with detergents to inactivate any residual material and to remove protein matter.
Thank you, Steve. Well, I guess at this point I'll end the webinar. Thank you all for joining us today, and have a pleasant rest of the day. Thank you.